As Claudio mentioned, so my name is Afshin Khalili, and I am product manager for the imaging reagents, for the 3D imaging, of course, the tissue clearing kit that we are offering at Miltenyi, and also the antibodies which are dedicated for whole tissue, whole clear tissue actually. And today with my colleague Luigi, we will go through the whole workflow of Miltenyi, what we offer for the 3D imaging. Try to change. Okay. Before jumping into the main story, I would like to just very briefly mention what we are offering at Miltenyi as for imaging. So on the right hand side, you see our a special biology platform called Maxima. With the dedicated antibody software and all the consumables actually, which is actually dedicated for the spatial biology and the high content imaging of the tissue slices. Oops. Can I go back? Yes. And here on the left-hand side, we see the light sheet microscope. This is actually the main story that I'm going to talk about together with the tissue clearing and the antibodies for 3D imaging. And we have also a very good idea of how we can combine these two platforms together, these two workflows together to have 3D imaging of the whole sample first, and then having the high content imaging and the spatial biology study of the tissue slices on the same sample of the same sample. But this is not the topic that I want to talk. We can discuss it of course later, but our focus is of course the light sheet imaging. So our talk has two parts. Firstly, I will go through the sample preparation and tissue clearing, and then Luigi will take over and talk about our light sheet system, which is called UltraMicroscope Blaze. Okay, I don't think that in this meeting I need to convince you why we need to do the 3D imaging. This is why I always have a slide. So if the question we want to answer is related to the 3D structure, then by 2D we just kill the data. This is obvious, and I don't want to waste time here convincing the experts why we should do that. Rather, I would like to just mention the difficulties that exist in the 2D and 3D sample preparations and the differences of the workflow. So 2D imaging, we have a sample like the mouse brain, for example, and it is opaque. We need to do the sectioning, physical sectioning using microtome, for example, and then antibody labeling, placing them on the microscope slide, and then doing the imaging, for example, in the confocal. This is good because this allows the microscopy of the opaque tissue, of course, with the sectioning. Imaging is easy, and there are tons of already established workflows around in each lab. It is not good because we compromise with the sample morphology, again with the sectioning. Then the 3D information is poor. The manual workload is really high, and of course, then at the end of the day, then it's really likely that you miss a specific part of the tissue by just sectioning out the interesting part physically, or not having the enough resources or enough time to do all the imaging for all the slides that you prepared, for example. And yeah. So then what if we do the 3D imaging of the large sample, doing it in one go to have a holistic view? This is what I mean. This is actually the human pancreatic carcinoma. This is just one example of what we mean by 3D imaging of the whole tissue, and I will show many more examples, of course. So in green, you see the tumor cells and infiltration of the CAR T cells in magenta. Each workflow, like 3D sample preparation workflow, has 4 steps. So the first step is immunolabeling, for example, using the antibodies. Secondly, we have clearing because the tissues are opaque. We need to clear them so that the light can pass through, and we have clearing. Thirdly is the imaging using the light sheet microscope, for example. And at the end, we have the data visualization and processing. So I will talk about the first two steps, and then Luigi will cover the last two. So firstly, immunostaining. 2D immunostaining, the classical immunohistochemistry, this is actually easy because you have a very thin slice, 5 micron, let's say, and then the antibody can easily penetrate. The incubation is usually a few hours, maybe even like the first one is like overnight usually, but that's even overkilling. What I want to say is that that's easy and already established. When it comes to the whole tissue, like mouse brain, then it is 100 times bigger. And the first issue that comes to mind is that this tiny antibody, this has hard time penetrate through the whole tissue homogeneously and then reach even the deeper side. So you can compare the size of the thickness of the slice with the size of the brain already. And on top, the tissue is opaque, so the light cannot pass through. You need to do the tissue clearing as well. Tissue clearing and immunolabeling, they both can interfere with each other. And then what remains is a very complex procedure, which is not easy to do. So all in all, the procedure is very complex. It needs a lot of time for doing the whole procedure, and background and irreproducibility of the data is also always there. So it seems like rocket science. This is what usually people tell me. It is not doable, but what I say is you just need the right tool, right material, and right protocols, and then that is as easy as cooking, depending on what you want to cook, of course, each cuisine. Okay. So this was about the immunostaining. Then the second part is the second step is clearing. As I said, the tissue is opaque. Um, so I keep it simple also here in this meeting. So the tissue is opaque because the light cannot pass through. It is being reflected, refracted, or scattered, absorbed by different type of molecules. So what we do in the tissue clearing is that we simply get rid of some of these molecules, plus we match the refractive index outside, inside, and everywhere in the tissue and the imaging environment, actually. That's why the light is passing through without being scattered or refracted, actually. So there is a very simple kitchen experiment. This is actually a glass bowl filled with these beads, and there's air in the middle, so the light is being refracted, so we can actually see them. Now the experimenter comes and pour water inside. What happens is that the refractive index is now very similar everywhere, and then the glass beads are appear to be uh, uh, how can I say that, like they are gone. So this is actually what happens uh, during the clearing. So this is the basic principle. Of course, water and glass, they have different refractive indices. This is just a kitchen experiment, just demonstrating the principle behind the tissue clearing. There are tons of ways and protocols for the tissue clearing methods available. I don't want to go through them, the hydrophobic, hydrophilic, hydrogel based. I won't go through them. Just if you want to learn more about this, then please just scan this QR code here, and then you will get this little booklet explaining the general idea of the tissue clearing. And this is not limited to what we have at Miltenyi. It's just very general about the tissue clearing concept. And just use the link, and then you will get the PDF. Or if you are in US, you get the hard copy. The hard challenges that usually researchers are reporting to us is the protocols are very hard to navigate. They're not like very good protocols available, easy to use. The procedures are complex, as I mentioned. The protocols are long and time consuming. Toxicity, reagent toxicity, this is one big problem. Some of the tissue clearings, they need special equipment. The GFP signal is quenched, for example. This is another big problem. And yeah, I'm trying to sort of address a solution for all of these issues that are reported. So let's go back to the first step. As I said, the first step is immunostaining, and at Miltenyi, we are offering the validated antibodies for 3D immunofluorescence. What is this? So these are actually the antibodies that we are offering. They are applicable for neuroscience and immuno-oncology. So mainly most of the markers that you need for your research are already available in different colors. And they are already validated for the 3D imaging of the large tissue. And this means less validation cost on your side. I don't know how much you are familiar with this process. So validation of the antibodies for 3D imaging, this would be very expensive, not only money-wise, but also time-wise, because it takes time until you test a specific antibody. And then it takes like, for preparation, it would take like, I don't know, about 2 weeks for mouse brain. And then labeling itself takes 10, 12 days, depending on your method. And then trying out different type of antibodies, of course, would be super expensive. So we have validated these in-house, and then this is less cost for on your side for the validation. These antibodies are already fluorochrome conjugated, so you don't need to do the primary and secondary antibody labeling. So one-time primary labeling, it would be sufficient. They are conjugated to VioDyes, which are the fluorophore from Miltenyi. These are tiny and also bright, tiny, bright, and very photostable. This would actually save time from your side, because then you need to do only one-time staining, not primary and secondary, already reduced to half, reduce the time to half. And also this would give you the optimal signal to background ratio. This I can also explain later. And these antibodies are recombinantly engineered, so we have also overcome the issue of the specific signal, and also the minimal background and maximum reproducibility. This is just one example by our antibody being tested by Gubra, the service provider in Denmark, testing our antibody on mouse model of Parkinson. This is the tyrosine hydroxylase antibody, which is labeling all the dopaminergic neuron in the brain. And they have tested this not with our tissue clearing kit, but with the standard iDisco clearing method. So I would not say anything, and then just please enjoy the nice labeling and travel through the dopaminergic world. Yeah, I also had the chance to talk to Jacob from this company, and yeah, nice saying that this is actually the best antibody that they ever used. Another example, again from Cupra, using this time our neurofilament and the healthy mouse brain, again using iDisco Plus clearing method. So yeah, this is just a point to mention here that our antibodies are not only validated for our own tissue clearing method, but also is compatible with other tissue clearing methods as well. Another good testimonial actually is coming from Chedotal's lab, Alain Chedotal in France, again using our neurofilament antibody on the human embryonic foot. And yeah, so here also you can appreciate the specific labeling of this antibody. Okay, just one example on what I mean by validation and optimization. So on the left-hand side here, you can see the labeling of the beta tubulin labeling of cerebral organoid, human cerebral organoid. And this is what happens when we have a non-optimized antibody. So this is a classical issue that the antibody is not penetrating deep inside the big tissue, but it stays on the outside of the tissue. When you have an optimized antibody for 3D immunofluorescence, then you have also a homogeneous labeling of the labeling of your target protein all over the tissue. About the tissue sizes, we can start from as small as 1 millimeter with the cerebral organoid, for example, coming to tumors, even bloody tumors, and then to the mouse brain hemisphere. The second step, tissue clearing. I can just mention what we have there. So this is the workflow for the tissue clearing, fixation, permeabilization, immunostaining, dehydration, usually happens in ethanol, and RI matching, and finally the 3D imaging. So for each part, we are offering a product. So we have the tissue clearing, max tissue clearing, which consists of the permeabilization solution, which you can use here, antibody staining solution together with the antibodies, and clearing solution, which you can use for the RI matching. The dehydration and fixation, this is normal procedure in every lab. Fixation is the PFA fixed tissues, and dehydration is usually in ethanol, which also exists in all of the labs. And finally, we also have a max imaging solution. This is the solution that you need to do the imaging with. This is the solution that at the end you immerse your sample in and do the imaging at the end of the day. So as you see, then we are covering the whole workflow from beginning to the end. And this is actually our standard workflow for the image. We are having also the different protocols for different purposes and applications, mouse brain hemisphere, blood-rich tumors, cerebral organoid, lung and intestine, liver, kidney, spleen, all mouse, also lymph node, and also we have a dedicated protocol for endogenous protein signal preservation, like GFP, for example, preservation in mouse brain hemisphere. Yeah, and this is how it works with the last step is the RI matching, the clearing. Actually, you can see in the course of 6 hours, mouse brain hemisphere on top and human ovarian carcinoma in the bottom become clear, and you can see through it. As I said, the protein signal preservation, endogenous protein signal preservation, this is also, this has a separate protocol on the same kit, using same kit with another protocol. So all of these protocols, actually, you can use the same kit with the for different applications using the different protocol, which is available on our website for free. This is actually very similar to our standard protocol that we have. Only the dehydration part, except of ethanol, is now happening in third butanol plus triethylamine. And then with this trick, you can preserve the endogenous signal. For the pigmented tissue, like mouse liver, spleen, kidney, embryo, mouse embryo, or even for the blood-rich tumors, the protocols also have one step of the depigmentation. Here is like no depigmented, and no, yeah, I'm saying the word correctly, right? A non-depigmented tissue and cleared. And here we see the depigmentation done with hydrogen peroxide, which is the classical way of doing that. And then in our protocol, our protocol is slightly different. We are using or recommending to use carbamide peroxide, which is less harsh to the epitopes. Yeah, so this you can also find on our website for the depigmentation. To sum up, yeah, this is actually the kit. The tissue clearing kit is not only tissue clearing, so it is a permeabilization solution, antibody staining solution, clearing solution, and imaging solution that you can later use for the imaging. This video I already showed how the tissues are becoming clear. So the clearing kit is this is highly efficient and robust. It is non-toxic, organic solvent based, optimized for the whole mount immunostaining. As I said, we have the dedicated protocols, and at the end, we also have a tips and tricks of tissue clearing. This is another document that you can also use for other types of for the troubleshooting in other types of organic solvent-based clearings. So it is not limited to our kit. Okay, I believe with this, then I am at the end of my part. I would like to hand over to my colleague Luigi. Luigi, the stage is yours. Sorry, I talked too much, so it's like less for you.
That is fine, no problem. Afshin, thank you very much. I see that I cannot share the screen as long as you are sharing, so I'm gonna steal the show for a minute. All right. Just give me a sign when you are able to see the slide. Yeah.
I think this
All right.
coming.
Yeah. As Afshin mentioned, it's not there yet.
No, it is there. It is there.
Okay. Thanks. As Afshin mentioned, in this few minutes, I will cover the last step or last steps of the 3D imaging workflows, which are represented by imaging, and as well as data visualization and processing. And in the case of our specific 3D imaging workflow, imaging is done with the UltraMicroscope Blaze, which is a light sheet microscope. Now, it was very convenient that there was another webinar on light sheet microscopy before ours, because then I don't have to explain all the principles of light sheet microscopy. Of course, our system also implements the so-called 90-degree principle. So illumination is perpendicular to the axis of detection, which allows you to excite only a very specific focal plane at a specific time point, which means that we don't get any out of focus fluorescence. The fluorophores are happier because they're not excited for nothing, and we don't have to take care of, again, out of focus light when it comes to detection. That's how our UltraMicroscope Blaze looks like, and the third dimension is given by the stage itself, which in our system is able to move in X, Y, Z. So what we are effectively doing with our samples during the imaging is just to move the samples up and down or in any direction which you would like through some static light sheets. And in particular, we have 3 light sheets per side, bidirectional illumination, to get rid of striping as well as shadowing as much as possible. Now, if we focus on our instrument, depicted in this slide here, you have its strength, or what we believe are the strengths of the system, the ability to image very large or multiple samples. The system is extremely easy to use thanks to a very intuitive software and a lot of automated function, dedicated light sheet optics, of course, and then we have sort of new feature, which is time to data. The microscope just got a lot faster than what it used to be, and I will cover that in a minute. Of course, another point that we also that we always like to mention is technical and applicative know-how. We are not just an instrument provider, but as Afshin nicely explained, we cover the entire workflow, and we are always happy to help users at any that require help at any of the steps of this 3D imaging workflow, no matter if it's staining or clearing or imaging. Now, when it comes to very large or multiple samples, our system is specifically designed for that. We do have very large imaging chambers, which, as you can see, would allow you to fit a sample as large as an entire mouse, which you see depicted here on the left. But also another way that we like to use this big space is to mount a lot of organs or samples in general and image them sequentially. In this video, it was just playing 5 organs imaging, but we have customers doing more than that. Ease of use, as I said, sample is very intuitive, plus a number of automated functions, some of which you see here. We have an automated objective exchanger. So as you see, the system comes with 3 objectives. You can move from one magnification to the next just via a simple click on the software. The system is able to do autofocus. This works on one channel, but also on multiple channels. And we have a nice automated sample release function, which basically happens at the level of the drawer. If you just pull the drawer to exchange the sample, the whole chamber will lower down and expose the stage so that you don't have to get crazy, emptying the chamber and refilling with again liquid or messing up your sample. It's actually very easy to work with. Yeah. All in all, this of course leads to easier handling of the system as well as reduced hands-on time. Actually, when it comes to automated function, the most powerful one is represented by the batch measurement. As I showed you before, you have a large chamber, so you can mount multiple samples. Via the software, you can also image them sequentially, right? So you don't have to, you just set the measurements once at the beginning. Of course, you can have different sample sizes or different channels because your sample might be completely different from each other. You do this setting up all at once at the beginning. You just press play, and then the microscope can run overnight and acquire as many samples as you like and save them in separate files, which hopefully you will find them the morning after when you go back to your lab. When it comes to optics, light sheet optics means both optics in excitation. So we have bidirectional illumination with 3 light sheets per side, but also in detection. Our objective lenses are quite unique. The microscope comes with 3 objective lenses, 1x, 4x, 12x. They are all high NA and low working distance objective in order to provide the best resolution, but still allow you to image something as large as an entire mouse that I've showed you before. I brought here a little example of the resolution which you could get. This is a 12x scan of a piece of a mouse brain. We are focusing on the cortex, and it's quite nice because you can appreciate all neuronal processes and get an idea of what would be the best resolution that you could get out of this system. As I mentioned, we have this new feature which are going into the direction of speed, and in particular, when talking about 2 developments. One is the so-called light speed mode. As the name suggests, this is just a different acquisition modality, which makes the microscope a lot faster. Just an example here for the imaging of a whole mouse brain, cleared obviously 2 channels, it would have taken 27 hours with the previous modality. So not the fastest, but on the other hand, you get a really good resolution out of it. With the new light speed mode, it would only take 37 minutes to get the whole brain, still 2 channels. And if you want to look at the quality, this is also not affected at all. So there's really just the advantage of being faster. As you can see by these 2 magnifications of 12x scan of this brain, there's absolutely, you definitely get cellular resolution, even when imaging with the light speed mode, which is really cool. The whole brain was a 4x scan. You have some more parameters below here. Again, as Afshin reminded a couple of times, you always find a lot of information on our website. Otherwise, we are always very happy to discuss further about it. The second development that we launched, and by the way, these 2 new products are on sale as of 2 weeks, is a dedicated software for the UltraMicroscope, the MaxIQ View 3D Large Volume. This software has been designed specifically to handle data coming from the UltraMicroscope Blaze. It reads directly what the microscope is acquiring, so you don't have to do any file conversion, no waste of time there. It's also able to do batch processing. So in particular, you have a series of algorithms that you could run your samples on: destriping, denoising, deconvolution, stitching, of course, and contrast compression. If you have your favorite pipeline, you could just set that up and apply into as many samples as you like and save the workflow for future applications, of course. It's user friendly. As I mentioned already, it doesn't require any data conversion. It has obviously a nice side-by-side viewer, so you can see once you have applied the algorithms what has changed, and if you are happy with that, because parameters can be a little bit tuned depending on your images. And the output, you have a couple of options when it comes to output, but the standard would be a TIFF file, so no crazy formats there. This should be able, you would be able to fit these data to any further software that you like. Yeah, it was a very quick overview. I hope I made it on time. Just wanted to remind you about our entire 3D imaging workflow, covering all the steps starting from immunostaining up to finalize and process images. I guess Afshin and I mean, I know about me. I guess Afshin is also very happy to take questions now. Thanks.